So first word about the exam. You should be able to see your scores now. I'll open up the answers here in another couple of days or so, probably tomorrow. I added 15 points to all scores, so that's where things stand. Could have added more, but I added 15. So we'll go from there, and then we'll don't forget, we have another exam in here next Monday. It goes pretty fast, then the final on Friday. So 2 weeks and it all comes to an end. Well, what we're going to do today is we're going to pick up the idea of the renal system, and we're going to talk about the structure and function of the kidney. And we'll first mention a few of the ideas of the function. And when you start thinking about what the kidney does for us, and it's one of these structures that we take for granted all the time, is running in background, and until things start to go wrong, we don't really pay much attention to it. But what it's doing for us is our kidneys are maintaining the elimination of nitrogenous waste. Every time we break down a protein, every time we break down amino acids, we end up with generating a little bit of ammonia. Now, ammonia is really, really toxic. Now, as long as we were single-celled organisms, there's so many things we've seen in here, this wasn't a real problem because the generation of ammonia would simply mean that ammonia would diffuse out of the cell into the extracellular environment. But what we find is that what we do in mammals is we carry that ammonia to the liver and we convert it into the structure called urea. Now, urea has a number of advantages. First, it's a means of getting rid of the ammonia and making it relatively non-toxic. Where ammonia is really quite toxic, urea is not particularly toxic. Now, you can't build up enormously high levels of it, but we're going to see as we work our way through here that the urea plays an important part in the function of the kidney itself, and so it can't be too toxic. When we start looking at other animals, though, when we start looking at reptiles and birds, we find in that case that the nitrogenous waste is mostly taken care of by something called uric acid rather than urea. And I'll show you a couple of drawings of that. But mainly we're going to be dealing with urea in here. One of the main advantages of urea is water soluble, so that means we can easily put it in solution in water and store this nitrogenous waste as urea in the bladder for hours until we eliminate it to the outside world. The other major thing that we find that the kidneys are doing for us is that they're regulating salt and water balance. And we know this more or less intuitively. We know it intuitively because if you go out and have a salty meal, you know it doesn't take very long before you become thirsty. So you start drinking water to begin to dilute that salt, and then over time you'll get rid of the salt. Also, if you drink a lot of water and take in a lot of water, that you'll then get rid of that in short order and bring everything back to balance. So there's a really important homeostatic maintenance function that we have in the kidneys, whether we're dealing with the nitrogenous waste or whether we're dealing with water and salt balance or whether we're dealing with pH regulation. And of course there's the erythropoietin that we talked about. We talked about the generation of erythrocytes. Now, once we've run through a little bit of a list of the kinds of things the kidneys will do for us, then we're going to move on into kidney structure. And the kidney structure that we're going to describe is fairly straightforward. We described earlier the amount of blood that goes into the kidney with every circuit of the blood coming out of the left ventricle. Something like a quarter to about 20 to 25% of the blood coming out of the left ventricle is now being passed through the kidney with every circuit through the cardiovascular system. So it's a tremendous blood supply to the kidney. And we're going to see that that blood supply is carried into the kidney by the renal artery. It's brought back out by the renal vein. But then, of course, in between the arterial inputs and the venous drain, there have to be a series of capillaries. Now, the vascular compartment in the kidney is fairly complicated, but the first part of it's straightforward. We're going to find that all of the blood going into the kidney goes immediately to a very thin veneer, a very thin layer called the renal cortex. That thin layer is no more than a couple of millimeters thick. It's on the outside edge of the kidney, and we'll talk a little bit about that. And we'll spend a lot of time in here talking about the renal cortex and the renal medulla. In other words, the very edge of the kidney, which is the renal cortex, and the medulla, which is the center. And we'll talk a little bit about that anatomy. But one of the major ideas that we'll point out early on is that all of the blood coming into the kidney is going out into the renal cortex, so that it goes from arteries to arterioles. But then those arterioles then will then feed blood into into something called the glomerulus. The glomerulus is really the capillary of the kidney. But here, the glomerulus is going to be a capillary that sits in a specialized structure called Bowman's capsule. And taken together, Bowman's capsule and the glomerulus is known as the renal corpuscle. So we'll talk a lot about how this is arranged, and we'll talk a lot about the blood supply. And we'll talk about how the glomerulus, in combination with Bowman's capsule, is the point of filtration. So what we're going to think about is the idea that the blood coming into the kidney is being driven into the kidney by the left side of the heart. That generates the pressure. And now we're going to find that out here in these glomerular capillaries, there's a considerable pressure against the wall. It's about 55 millimeters of mercury. So we have this considerable pressure against the wall of the glomerulus, and we're going to find then, just as we talked about in generating lymph, that there are two major pressures involved here. An outward pressure, which is going to be mechanical hydrostatic pressure, and there's also then going to be an osmotic pressure, which is drawing the fluid back into the blood vascular compartment. But in addition to those pressures that we talked about in the generation of lymph, we find also in this region we have another pressure that's generated by the pressure of Bowman's capsule. We're going to add all of those together, but in the long and the short of all that is there's going to be a net outward pressure. And net outward pressure is now, because there's a delta P, is going to generate a flow. And so now we're going to get a flow from the blood vascular compartment into the renal compartment. And then that fluid moving from the blood vascular compartment into the renal compartment is going to be now drained through the nephron, the collecting duct, the renal calyx, the ureter, the bladder, the urethra to the outside world. Now, if you look at this list, you'll then begin to recognize that this is a classic arrangement for an epithelium, because we can follow the cells lining all of these structures all the way back to Bowman's capsule. And that will be a continuous layer of epithelial cells. And it's a true epithelium because it connects to the outside world. So we'll lay out this anatomy. And then what we'll begin doing is we'll be talking about the forces that are generated in filtration and how these forces are carrying small molecules from the blood vascular compartment into the renal compartment. Now here's where we'll begin discussing the interesting arrangement of what the kidney does. It seems woefully inefficient, but it's the way it functions. And that is, it forces out enormous amounts of water and small molecules from the blood vascular compartment into the renal compartment. And then there are all these transport mechanisms then that pick and choose which of these molecules will keep and which molecules will get rid of. So it's this great sorting system. A sorting system where the pressures are moving small molecules from the blood vascular compartment to the renal compartment, and then we're going to have a whole series of events going on among the epithelial cells in the renal compartment to pick and choose what we want to keep and what we want to get rid of, and we'll walk our way through that. One of the ideas that will come to the fore here is that even though we have some million nephrons in our kidneys, and each one of them only moves a very small volume of fluid, that in combination, these guys are moving about 125 mils of fluid. About 125 mils of fluid is moving from the blood vascular compartment into the renal compartment every minute. Now, you know full well you're not going to send that much fluid to the outside world. After all, 125 mils per minute for 60 minutes is close to 7 liters, and so that's a mighty full bladder. So that tells us then that there's reabsorption of these contents. The molecules and small molecules, the small molecules in water that we have forced from the blood vascular compartment into the renal compartment are now going to be reabsorbed. The first site of reabsorption, and what we'll talk about today, is the proximal convoluted tubule. In the proximal convoluted tubule, we'll find that some 60% of the sodium in the water that has been forced out of the blood vascular compartment into the renal compartment will be resorbed at this point. The rest of it's going to pass through the rest of the nephron, and we'll talk about its fate there. Then amino acids and glucose. The amino acids and glucose in the proximal convoluted tubule will almost entirely be reabsorbed. It's one of these efficient systems where 5 millimolar glucose is going to go from the blood vascular compartment to the renal compartment, and within, a few millimeters, running through that proximal convoluted tubule, the transport systems will reabsorb 100% of the glucose, the amino acids. And we'll talk about how that's done through these molecules called sodium-driven cotransporters. Oh, and I should put another set down here. The facilitated transporters. Well, let's just make a short list, though. Start here with functions of the renal system. And as we mentioned just a moment ago in the introduction, one of the main functions is disposing of nitrogenous waste. After all, anytime we break down proteins, anytime we break down amino acids and break down amino acids from proteins, we end up with a waste product of ammonia, which is NH3. Now, ammonia itself is pretty toxic. And again, we can go back to when we were thinking about the cardiovascular system, and we said as long as we were individual cells, or just thin sheets of cells as in sponges, there wasn't too much of a problem here. We could simply use diffusion to allow ammonia to leave the cells and go to the outside world. But once animals become three-dimensional creatures, once you begin to see the three-dimensional shape of an animal, then we're too far away from the outside world to accomplish these tasks by simple diffusion. So now what we need to invoke is a combination of the cardiovascular system and renal system. The cardiovascular system first plays a role in that it moves ammonia to the liver. And then in the liver, ammonia, through something known as the urea cycle, ammonia through the urea cycle is converted to urea. Now, I'll never ask you to recapitulate this structure, but I think it's worth taking a moment to look at it. Urea is a single carbon with a double bonded oxygen, and then two nitrogens with their hydrogens. Now, this end of the molecule is going to be charged, and this end is going to be polar. So what I want you to recognize from this structure is this is highly soluble. Because of its charged and polar nature, you can put a lot of urea into solution. And we're going to use that as an important function in the kidney. But the other functions that we see for the kidney are to regulate blood pH. Now, the blood pH regulation we talked about the other day was in terms of the use of bicarbonate, and how bicarbonate was the essential buffer in blood, and how we would use bicarbonate to maintain pH in the blood between 7.2 and 7.3. Well, we find that another point of regulation of pH in the blood is here in the kidney, and what we'll do is that we'll basically move hydrogen ion from the blood to the renal compartment. And then normally, under most circumstances, we find that urine is acidic. This is telling us that we're taking excess hydrogen ion out of the blood, putting it into the urine. It's being buffered in a number of ways along the route, but basically we're using the hydrogen ion in the blood, which now is lowering the pH of the blood. In order to get rid of that hydrogen ion, we use the kidney and move it out into the urine to the outside world. The third important function of the kidney is to regulate ion and water balance. Now, if you take another course in physiology, you'll delve down into some of the really intricate mechanisms to control things like calcium and phosphate and some of the other ions. But we're going to deal mostly here in terms of sodium and to some extent chloride. But along with this, we're also going to be talking about the regulation of the amount of water. In blood. Now, that's a roundabout way of saying that what the kidney is doing is it's regulating ion concentrations. In the blood. We don't want to dilute those ion concentrations too badly. We don't want to allow those ion concentrations to get too high. And so what we find that we're doing here is that we're functioning to regulate both the amount of water and the amount of ion. Now, we know this intuitively. We know this intuitively because of just everyday behavior. And so if you go out tonight and you go out and have a really salty meal, you go get a really tasty pizza, you know that it's only going to be a couple of hours or maybe even not very long at all. You become thirsty because you've taken in all this extra salt. And then you take in extra water to compensate for taking in that salt. But now the first step is that your ion concentration in the blood has gotten too high because you're taking in too much sodium. So then you drink some water to try to dilute that. But then there are these compensatory relationships between the kidney and the blood, which is now going to bring those back into balance, so that we always have more or less 130 millimolar sodium ion in the blood, in the extracellular space. So even if we take more water in, we take more salt in, we regulate through the kidney this concentration in this range. Now, there's some delay in this, and we'll talk a little bit later about why there is a delay, but you also know that if you go out and have a salty meal, sometimes you'll get up in the morning and you'll find your rings are just fitting a little tighter than they used to. And that's because once you've taken the sodium on, water has followed. Well, as we move through this, recognize that water is going to follow salt. And so if you've had a very salty meal, taken a lot of salt, then that sodium then is going to act as an osmotic draw, and it's going to pull fluid into the extracellular tissues. And then that's going to cause some swelling, particularly in your hands and feet. And so that's largely due to this movement of water into the extracellular space. And then finally, there's the character here that we've talked about before, whereas the kidney secretes erythropoietin. To to regulate red blood cell production. So those are basically the functions that the kidney takes care of for us. And again, it's one of these things we just assume works all the time. But renal disease, kidney disease, is a serious problem of public health. And lots of people end up getting renal disease. And it's a chronic issue that is dealt with over years and years of people's lives. So with that, let's now think about the blood supply of the kidney. And on this panel, we're going to talk about the anatomy of the kidney and its blood supply. And we've drawn this before when we were talking about erythropoietin. We drew the two kidneys and emphasized that those two kidneys had a tremendous blood supply because the renal arteries branched from the descending aorta. So these renal arteries supply the kidney. The rules of the closed system that we talked about when we talked about the cardiovascular system apply here as well. And we'll see that the blood vessel, the artery coming in, branches and branches and branches. Now, although I'm not doing it very artistically here, what I'm trying to convey is that the artery, the renal artery branches into arterioles. And these further branch to supply the capillaries. So this is the same pattern we've seen every place else. The arteries branch into arterioles. The arterioles then feed those tiny structures which are composed of a single layer of endothelial cells called the capillaries. So they branch to supply the capillaries. But now the capillaries have a special name in the kidney. These are called the glomeruli. Singular is glomerulus. Now, I'll show you a picture a little later, but when we think of the glomerulus, when we describe these, we describe them as a tuft of capillaries. But when I draw it up, I'll just draw it as a single tube because that'll help us understand how fluid is going to move from one compartment to the next. But in fact, it's a group of capillaries. And these structures up here, all of the blood, one of the major ideas of the renal artery and its branches is that all blood to the kidney goes to the renal cortex. Well, here's this term again. We've seen the term cortex over and over again. And remember, the cortex is simply a thin outer layer. Whether we're talking about the cerebellar cortex or the adrenal cortex, it's the outer layer. Well, here in the kidney, there's this outer layer just a few millimeters thick. It's out towards the edge of the kidney. This is called the renal cortex. And the renal cortex is the destination of the blood that's flowing into the kidney through the renal arteries. So that now the blood is not bothering to stop or send or branch into capillaries at any point down here in this region I'll show in blue, called the renal medulla. So in other words, the blood contained within the arterioles is crossing the renal medulla and going all the way up here to the renal cortex. There's almost no blood supply in terms of capillaries in the renal medulla. It's all going to the renal cortex. And if we look at this structure, we find that the destination of the blood coming into the kidney is to flow to these specialized capillaries, the glomeruli. But the glomeruli are tucked into a structure called Bowman's capsule. So what I'm going to do with this panel is I'm going to take this panel and describe it as the renal corpuscle. The renal corpuscle as the interface between the blood vascular and renal compartments. The renal corpuscle is composed of glomerulus and a structure called Bowman's capsule. This is blood vascular. This is renal. In fact, we're going to see that Bowman's capsule is composed of epithelial cells. These are epithelial cells called podocytes. Well, let's see if we can make a crude drawing of this. Now, Bowman's capsule, I'm going to draw it this way. This is meant to be Bowman's capsule. Because as I said in the introduction, the inside of Bowman's capsule connected to the rest of the nephron and to the collecting duct to the outside world. These will be true epithelial cells because that's going to be a continuous layer of cells that go to the outside world. So we can draw in here these epithelial cells. Now, because they are epithelial cells in Bowman's capsule, they get a special name. These are called podocytes. Now, nestled in here, and I'm going to show this as a kind of oxbow, but in fact it's going to be a fairly complex network of capillaries. The structure that I'm trying to generate here is known as the glomerulus. Now, the glomerulus, being nothing more than a capillary, is lined by a layer of endothelial cells. So these are the endothelial cells These are the endothelial cells of the glomerulus. Now there's blood flow through here. This is an extension, remember, of the arterioles, and so there is blood flow through here, and I'm going to put some arrows to signify the direction of flow. The structure down here is said to be the the structure down here is said to be the afferent arteriole. Back to the old nomenclature, afferent to carry toward. So this is the tube, this is the arteriole that's carrying blood into the glomerulus. But on this side, we're going to see that there's also a drain. So there's blood flow through, and now blood flow being carried away from the glomerulus is being carried away in this structure known as the efferent arteriole. So again, we follow the blood coming into the kidney. It comes into the kidney and the renal artery, and then all of that big artery then branches and branches and branches in these tiny arterioles. And these arterioles then, in turn, feed afferent arterioles that go into the glomerulus, and efferent arterioles are going to drain those specialized capillaries, the glomeruli. Now remember too, and we'll come back to this a little later, that whenever you see this term arteriole, remember that these are surrounded by smooth muscle. Well, the rest of this, what we're going to start talking about now, is something known as glomerular filtration. Glomerular filtration is just a fancy word that says movement of ions, water, and small molecules from the blood vascular compartment to the renal compartment. Well, that's a mouthful, but let's think a little bit about how that's going to happen. So remember, this is a classic capillary. There is a pressure on this capillary. Remember, we talked about lymph. We said that there is a hydrostatic pressure. There's a pressure that's generated by the contraction of the heart, the force of the muscle in the heart to produce a hydrostatic pressure against the fluid volume. That hydrostatic pressure extended all the way from the heart out into the arteries, the arterioles, and into the capillaries. Well, that's true here as well. We now have a hydrostatic pressure. The glomerular hydrostatic pressure can be measured, and it's about 55 millimeters of mercury. So that's an outward hydrostatic pressure against the wall of the tube. That's going to be trying to move fluid and small molecules between the adjacent endothelial cells. Now, when we've talked about the generation of lymph, we emphasize the idea that the individual endothelial cells were separated one from another by narrow gaps. These are meant to be the gaps between adjacent endothelial cells. And remember when we talked about the spleen, we said the spleen was really unusual because it had gigantic gaps between adjacent endothelial cells. When we talked about the brain, we said that those gaps between adjacent endothelial cells were very small and were stitched together by tight junctions. Here we have something in between. We have gaps that are fairly large but not enormous. They're large enough that they will basically pass. These gaps are large enough to pass molecules with a molecular weight of more or less 1,000. So that means ions and water and ATP and glucose and amino acids will freely move from the blood vascular compartment into the renal compartment. But what we also find, though, is that there are a set of pressures in here we have to think about. The first pressure is this glomerular hydrostatic pressure. But then, and you'll remember this from the story of generating lymph, is that there's now going to be an osmotic pressure. Because even though the size of the molecules that can pass through between these adjacent endothelial cells is in the neighborhood of 1,000, that means molecules bigger than that can't pass. And if you look at something like albumin, the molecular weight of albumin is in the neighborhood of about 40,000. So that means the proteins in plasma can't move. They're stuck on the inside of that tube. So even though we're forcing out water and small molecules, we're leaving behind protein. That protein generates an osmotic force. Because we've forced the water out into this compartment, we have left a lower concentration of water inside the glomerular capillary than we have out here in Bowman's capsule. Because the water concentration is less here than here, there's a force bringing the water back into the capillary. And this is said to be the osmotic force. And the osmotic force is about 30 millimeters of mercury. And again, I won't ask you really to hold on to these numbers, but what I want to do is work through this series of numbers to generate a delta P. Because we also find that as we fill Bowman's capsule, and you can imagine Bowman's capsule as a kind of balloon that's being filled with fluid surrounding that capillary. As that balloon fills with water, the pressure now begins to build, and we'll also get a pressure moving in this direction. There'll be a pressure moving in this direction called the pressure of Bowman's capsule. This is a hydrostatic pressure. It has about 15 millimeters of mercury. But now let's put all this together. You'll note that there is a pressure of about 55 millimeters of mercury going from the blood vascular compartment into the renal compartment. But when you add together the 30 millimeters of mercury going in the opposite direction, which is osmotic, and 15 millimeters of mercury, which is Bowman's capsule, That's also going from the Bowman's capsule back into the blood. If you add these two together, that's 45. So you have 45 millimeters of mercury pressure moving fluid from Bowman's capsule back into the blood, but you have 55 millimeters of mercury moving moving fluid from the blood vascular compartment into the renal compartment. In other words, we have a net pressure of 10 millimeters of mercury moving fluid from the blood vascular compartment, let me spell it out, moving fluid from the glomerulus into Bowman's capsule. This is a long-winded way of saying that we have a delta P. A delta P of about 10 millimeters of mercury. When you have a delta P and a resistance, you have a flow. So we're now having a flow of fluid from the blood vascular compartment into the renal compartment. And although for each individual Bowman's capsule, this flow is tiny, when you add together the flow in all million or so of the Bowman's capsule, you get a flow that tends to be in the neighborhood of about 125 milliliters per minute. So we're moving about 125 mils per minute from the blood vascular compartment into the renal compartment. But if you simply would imagine that all of this fluid moving from the glomerulus into Bowman's capsule is going to go to the outside world, you see that can't be true. Because if we imagine sitting here for an hour, times 60, this is 0, 0, carry the 3, 15, that's 7.5 liters per hour. That's the better part of a gallon. So we simply cannot possibly be moving that much fluid from the blood vascular compartment through the renal compartment and out to the outside, out into the bladder in that period of time. We've got to be doing something about reabsorbing those fluids. So any questions to here? Well, let's take a moment then and summarize where we've been and talk a little bit about some of the molecules that are important. Now, again, I'll never ask you to memorize these structures, but I think it's instructive to take a moment to look at them, where we have ammonia, which is highly soluble but is toxic. Urea, which is highly soluble but not very toxic, but now what we've been able to do is take this ammonia and attach two ammonias onto the carbon, have this double bonded oxygen, and this provides a nice soluble molecule. This molecule over here is the waste product that we see in birds and reptiles. It's not especially soluble, and birds don't have a bladder. Birds take the output of their kidneys and dump it directly into the cloaca, which is really the large intestine. And then it goes to the outside world. So their nitrogenous waste goes out with the feces. That's the way it's blended together in the cloaca. And this is a more complicated molecule than urea. And it has a problem of not being especially water soluble. We generate some uric acid as well. And it's a byproduct mostly of breaking down nucleic acids, the RNA and DNA. And so when we're on a high meat diet particularly, we tend to produce large amounts of uric acid. One of the shortcomings of our inability to handle uric acid very well is we'll sometimes build up uric acid in the joints. When you begin to get deposits of uric acid in the joints, it's extremely painful, and it's known as gout. So people who have gout, in fact, have this problem of depositing uric acid in their joint capsules, and they end up with these crystals in the joint capsule, and it can be quite painful. This molecule over here is creatinine, and it's one of these molecules that many of you, particularly in exercise and health sciences, will recognize. What creatinine is it's a waste product of creatine phosphate. Creatine phosphate is a storage molecule where we store phosphate in muscle. And it's one of the places we turn to grab phosphates to regenerate ATP when we're exercising hard. And so creatine phosphate is this storage molecule for readily available phosphate to make ATP. What the creatine phosphate does, though, once we pull that phosphate off of it, is we end up with this waste product called creatinine. If you've ever looked at a blood panel that you have after going to the doctor, one of the numbers that you'll see is a level of creatinine. And if you take another physiology course, you'll spend some time on this, because creatinine ends up being an important indicator. It's not a great measure, but it's a rule of thumb indicator of kidney function, because creatinine easily moves from the blood vascular compartment into the renal compartment. It's what they call readily filtered from the glomerulus into Bowman's capsule. But creatinine then is not reabsorbed. It passes out into the renal compartment and goes to the outside world without being reabsorbed. So if you have high levels of creatinine, it indicates there may be a problem with kidney function, and that you're not moving these small molecules from the blood vascular compartment into the renal compartment as readily as you should be. So high creatinine levels are an indicator of kidney problems. This is just an anatomy figure, and if you spend some time in BMS300, which is gross anatomy, you'll spend a whole lot of time looking at the abdominal cavity and looking at where things are placed. These kidneys are said to be retroperitoneal. They're behind the peritoneum. They sit up against the body wall. All I want to really recognize here is a couple of things. First, these sort of yellow things on top, those are the adrenal glands. Ad meaning on. So those are the adrenals. Well, here, these are the kidneys. Sometimes they're known as the epinephros. Again, using the term rather than renal, nephron, epinephros, on the nephros. So these are the kidneys here, these sort of fist-shaped structures. And what it's showing is the blood supply coming into the kidneys. And now it's showing the continuity between the kidney of something here called the renal calyx into the ureters, which are draining the fluid from the kidney and into the bladder. The fluid now moves into the bladder, is stored for some time in the bladder. And then at a chosen time, we can use the smooth muscle of the bladder to express fluid from the bladder to the outside world through the urethra. And again, this is one of the advantages of mammals being able to use the urea, which is highly water soluble. We can store that urea solubilized in water in the bladder. But one of the ideas that we're going to run into is that if you follow the epithelium of the urethra into the epithelium of the bladder, into the epithelium of the ureters, and then into the epithelium of the renal calyx, then you're going to follow that epithelium right back into the collecting ducts and into the nephrons. Just saying that we have a continuous layer of epithelial cells from that point in the renal corpuscle, where the glomerulus meets Bowman's capsule. We have a continuous layer of epithelial cells from that site to the outside world. This is a figure that's trying to get at the idea of the blood supply. And what it's trying to show is the renal artery coming into the kidney and then showing these branches of all of these all of these arterioles. But the concept here is that all of these branching arterioles are going to be feeding blood into this very thin layer called the renal cortex. The medulla is going to be mostly involved with the actions of the kidney itself. But what we're going to see here is the blood is all going to the renal cortex, and then those arterioles are breaking in or flowing into the glomeruli. Again, another figure trying to represent the branching structure of the blood coming in through the red arteries and arterioles, and the venous structure, which is draining the blood from the kidney. Now, what this figure shows that the others don't are a couple of things. First, it's trying to convey that there's this thin layer out here called the renal cortex. It's trying to show that the blood is flowing into the kidney and the blood supply is going all up to the renal cortex. And then it's showing these structures known as the renal pyramids. Now, we're going to come into that a little later. Those renal pyramids are almost entirely composed of branches of the nephrons. But the other piece that it looks at here is how this renal calyx feeds into the ureter. And the renal calyx now is going to be like a funnel. And it's going to be collecting fluid, which empties from 500,000 or so, well, not that many, empties from several thousands of collecting ducts into the renal calyx. So what we're going to see then, let me do this first. So what we see here is a diagram representing the renal corpuscle. So what I depicted as a simple flow-through, a simple sort of oxbow flow-through, what this is beginning to depict in much more detail and in a more realistic nature is this tuft of capillaries. This is a sort of tortuous route of capillaries in here that are now surrounded by this structure of Bowman's capsule. So again, this is the interface between the blood vascular and the renal compartments. Blood is flowing into the glomerulus through the afferent arteriole and is being drained through the efferent arteriole. And then in this region we have the blood contained within the glomerular capillary and separated then by the endothelial cells lining the renal capillary from the fluid out here in Bowman's capsule. And again, this onion-like structure, those pebble-like structures depicted in Bowman's capsule are meant to be the epithelial cells, the so-called podocytes. And so those are going to be the podocytes that are forming the surrounding of the Bowman's capsule. Then this figure, this highly schematized figure, is trying to describe the blood flow into the system and the various pressures that are involved. This is where we just closed with, where there's blood flow from the afferent arteriole into the glomerulus. There's a hydrostatic pressure of about 55 millimeters of mercury. But in the opposite direction to the movement of fluid from the blood vascular compartment into the renal compartment generated by the hydrostatic pressure, there's now an osmotic pressure that's pulling fluid back in. And there's also a hydrostatic pressure of Bowman's capsule, which is pushing fluid back in to the glomerular capillary. But when all is said and done, there's a net pressure outward that's moving fluid from this series of tubes out into Bowman's capsule, and we'll go into the next step called the proximal convoluted tubule. This is where we're headed. Where we're headed here is to describe the structure of the nephron and follow the fluid through the nephron and describe at what points we reclaim and don't reclaim the small molecules. And this is a pretty picture, and I'll simplify it a lot when we draw it on the board. But again, here's this onion-like structure of Bowman's capsule. This is the tuft of capillaries, the glomerulus sitting in Bowman's capsule. This is going to extend into the proximal convoluted tubule. It's got this sort of convoluted structure to it. It winds back and forth. That's the proximal convoluted tubule that now leads into the descending limb of the loop of the nephron, the ascending loop of the nephron, out to the distal convoluted tubule that drains into the collecting duct. That's basically where we're headed now. We're going to walk our way through these regions of the renal compartments, walking our way through the fate of the fluid as it moves into the proximal convoluted tubule, the proximal convoluted tubule, the descending limb, the ascending limb, distal convoluted tubule, and the collecting duct. And then at the end, we'll talk about the blood vessels that are surrounding the structure called the vasa recta. So any questions to there? If not, let's take a break Come back in a few minutes and we'll start talking about what's going on in the proximal convoluted tubule.